In today's video, I'm going to be talking to you about photosynthesis. Photosynthesis is one of the most incredible metabolic reactions that you will learn about. Imagine if when you got hungry and you needed some sugar to go and do something like grow, instead of having to go down to the shop and buy some food and cook and eat it and digest it and absorb it, take it into your cells, you could just go lie out on the lawn and the sun's rays would hit your cells and your cells would use the sun's energy. They'd use a little bit of carbon dioxide, a little bit of water, and they'd make sugar. It's magic. Photosynthesis is a process used by plants, algae, and cyanobacteria to convert sunlight, water, and carbon dioxide into a long-term energy storage in the form of sugar. That sugar can then be used to be converted into other biological molecules like amino acids and fatty acids and other types of carbohydrate, or it can be used in respiration to release energy for cellular functions. Photosynthesis is split into two stages. We're going to just talk about the first stage today. The first stage of photosynthesis is known as the light-dependent reaction, and it occurs on the thylakoid membrane. The second stage of photosynthesis is known as the light-independent stage, and this occurs inside the stroma. When we talk about the light-independent and light-dependent reactions, sometimes when we just look at one of them in isolation, it can be hard to entirely see the purpose, but it's really important that you understand that everything that occurs in the light-dependent stage is there to create two things. It's there to create a reduced coenzyme called NADPH and to produce ATP. Why is it producing those things? Because these things are needed in the second stage and they will be used to produce glucose. So the first stage, the light-dependent stage, is there in order to create these two substances, which will then travel off into the stroma and be used to create glucose. So it's really, really important that you bear that in mind from the very beginning. You might think to yourself, why have I just made this one molecule of ATP? Why have I made this reduced coenzyme? They don't seem very useful to my cell. It's because they're going to be used later on. So let's talk about the process. So here we can see our chloroplast, and if we just focus in, we can see where the light-dependent reaction occurs. So the light-dependent reaction occurs across the thylakoid membrane. So it occurs here. So let's focus on that area for a minute. So let's first draw our thylakoid membrane so we can put it into context of where it is. And then let's draw the embedded carrier proteins and ATP synthase molecule. So this process of producing ATP and reduced NADP, which is a really important coenzyme. Remember, you learned about coenzymes when you did respiration. So this is the same sort of molecule. It's a coenzyme. Its job is to carry hydrogens to the next stage. So this process is known as photophosphorylation. Using light to phosphorylate a molecule. And in this case, we are going to use light to phosphorylate an ADP molecule to produce ATP. So in actual fact, the version of photophosphorylation that I'm going to talk to you about now is a little bit simplified. It's at the level that you need to know for your specification at A level. In actual fact, there are two types of photophosphorylation. There is cyclic photophosphorylation and non-cyclic photophosphorylation. And if you read other material, you can read about both of those, but we're just going to talk about it at the level that you need to know about for now. So let's take a look at our electron transport chain that we have here in the thylakoid membrane. Take note of where the thylakoid space is, so the space in between the two thylakoid membranes. So we can see that if for a minute that we were just to look back at our chloroplast and have a think about where we are. So here is our thylakoid space. and here is the stroma. So the thylakoid space is in the center here of the thylakoid between these two membranes. So we've got this membrane that's in sort of an enclosed circle, and then we've got the thylakoid space that's also filled with fluid, and then we've got the stroma on the outside. So it's really important that we understand the geographical location of where this is occurring. So the thylakoid membrane and the thylakoid space and the stroma, this is where it is. And we can see this blown up version of it here, where we've got the phospholipid bilayer, and then we've got our transmembrane proteins, and we've got our ATP synthase. And we know that the function of ATP synthase is to produce ATP. So let's just focus first on this antennae complex here. So this transmembrane protein, which is also known as an antennae complex, which contains many chlorophyll molecules. So if we were to just think about that for a minute in terms of the structure of that molecule, it looks sort of like a cone shape. And embedded in this cone are many, many chlorophyll molecules with their little heads poking out and their little tails trapped in the membrane. So obviously this is a really simplified drawing, but you're going to have to use your imagination, okay? So light is going to hit this transmembrane protein, and the energy from the light is going to excite a pair of electrons from the magnesium ion at the center of this chlorophyll molecule. So these pairs of electrons will leave the electron outer shell of this magnesium ion, and they will be donated to the next chlorophyll molecule, and they'll just get passed down, 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 all the way down to the center, to the bottom here, where you have a chlorophyll molecule. And this chlorophyll molecule will lose its pair of electrons, and this pair of electrons will get donated to the electron carrier, which is in the thylakoid membrane. This process of losing a pair of electrons from your chlorophyll and donating them to the first electron carrier is known as photoionization. So photoionization is that loss of a pair of electrons from your chlorophyll molecule. So when light hits it, that excites the electrons. And these pair of electrons, they leave the outer shell of the chlorophyll molecule, and they go to the first electron carrier in the thylakoid membrane. So you might just want to take a moment to remind yourself of the structure of a chlorophyll molecule. So we know that a chlorophyll molecule, it has a porphyrin ring as its head, and in the center of the porphyrin ring, it has its magnesium ion, and then it has this hydrocarbon tail. And we know that the hydrocarbon tail is embedded in the membrane, and then the head sits on the surface of the membrane. So let's just go back here. So what's happening? So light is going to hit our antennae complex, our transmembrane protein containing chlorophyll, and that will cause this to lose a pair of electrons, which will get donated to the next electron carrier. As these electrons move from one electron carrier to the next, this is a series of redox reactions, because one is being oxidized and the other is being reduced. As the electrons move from electron carrier to the next, they move down their energy gradient. And as they move down their energy gradient, the energy that they release is used to actively pump protons into the thylakoid space. So protons are pumped up into the thylakoid space. So you can see down here that it says in the stroma there's a low concentration of protons, and in the thylakoid space there is a high concentration of protons. There is two things that causes this high concentration of protons to build up in the thylakoid space. And we're going to talk about each of them in turn. The first is the movement of the protons from the stroma up into the thylakoid space via active transport. Using a proton pump. And this active transport is fueled by the energy released by the movement of the electrons from one electron carrier to the next. The second is through a process known as photolysis, photolysis. So just take a minute to think back to the photosynthesis equation. We know that we need carbon dioxide and water and a little bit of light to make glucose. And that carbon dioxide is going to be used later on in the next stage, but this is where the water comes in. So in the thylakoid space, a water molecule is split in a process known as photolysis. So water is split into 2 protons, 2 electrons, and half an oxygen molecule. This oxygen molecule is excreted as a waste product. The 2 electrons are used to replace the lost electron from the chlorophyll molecule, returning it back to its original state. The pair of protons are released into the thylakoid space, and they remain there in order to build up a high concentration of protons in the thylakoid space. So, now we've got this high concentration of protons in the thylakoid space, lower concentration in the stroma, so we have created this concentration gradient. We know that protons cannot move directly through the phospholipid bilayer because they are charged, so they need to move through a channel. They move through the ATP synthase channel via facilitated diffusion. This process is called chemiosmosis. So they move via chemiosmosis. As the protons move down the ATP synthase channel, this creates something called proton motive force. This causes the ATP synthase to move, to change its conformation, and as it does, it causes the ADP and the inorganic phosphate to bind together and form a molecule of ATP. The proton is released into the stroma. So now we've got to solve the problem of what happens to these electrons and these protons. So the electron needs to, of course, leave this electron transport chain. Now, unfortunately, I haven't really left myself enough space. We're just going to have to improvise. Also embedded in this membrane is a big enzyme complex, which is known as NADP reductase. NADP reductase, you might have already guessed it, reduces NADP. NADP is a coenzyme. The protons released from the ATP synthase and the electrons released from the electron carrier in the electron transport chain combine together to form hydrogen, and these join with NADP to produce NADPH plus H+. This reduced coenzyme then travels to the next stage of photosynthesis, which we will learn about in a bit. So let's just recap the key ideas. Light hits our transmembrane protein containing chlorophyll. That chlorophyll loses a pair of electrons. Those electrons travel along the electron transport chain and release energy to pump protons from the stroma into the thylakoid space. At the same time, photolysis is occurring, which is a reaction which breaks down water to release a pair of electrons which replace the lost electrons from the chlorophyll molecule and to release protons into the thylakoid space. This builds up a high concentration of protons in the thylakoid space. These protons move via facilitated diffusion or chemiosmosis through ATP synthase into the stroma. As they move through, they cause the formation of an ATP molecule. When they are released in the stroma, They combine with a pair of protons released from the electron transport chain and form a hydrogen atom which reduces NADP, a coenzyme, nicotinamide adenine dinucleotide phosphate, a coenzyme involved in the photosynthesis reactions. This is catalyzed by the enzyme NADP reductase. So by the end of the light-dependent reaction, we have created ATP and reduced NADPH. These will then go to the second stage of respiration, where they will be used to make glucose. What new words have you learnt? You've learnt photoionization, so the use of light to ionize the chlorophyll molecule and cause it to lose a pair of electrons. You've learnt photophosphorylation, the use of light to phosphorylate the ADP molecule. Photolysis, the use of light to split a water molecule. And I think that's probably it. Well done.